tryptophan operon regulation at transcription initiation. This is again a very interesting operon system that talks about one of the phenomenon of regulation of gene expression, and that is regulation by repression. So let us look at the learning outcomes of this topic. So the tryptophan operon regulates the biosynthesis of tryptophan, and it has 5 structural genes for enzymes that structure the pathway. So unlike the lac operon, which is a catalytic pathway where the degradation of a component is being taken care of by the operon, in this case the operon is taking care of a biosynthetic pathway. That means a product is being formed. Now, the second learning objective is that the tryptophan operon is regulated at the initiation of transcription by a tryptophan aporepressor. So, when the repressor is being synthesized, it is synthesized in the form of an aporepressor, and so it is inactive basically, but that gets activated when it binds to the corepressor, and in this case it is the tryptophan itself which acts as a corepressor. The concentration of tryptophan therefore plays an important role in repressing the operon, which is a switched on mode. That means basically the operon is on an on mode, that it is switched on, but depending on the concentration of the tryptophan, it can get switched off. So in a way, the product of the operon is regulating its own synthesis. Now, this is definitely an operon that is an example for control by repression. And in this case, in the tryptophan operon, the repression itself is responsible for an 80-fold decrease in the synthesis of tryptophan. So it is able to repress the operon by 80-fold. Just looking at the fact that the entire system that is formed due to the operon is what is called as tryptophan synthase system. So there are 5 genes that give rise to 5 different enzymes, and each of these enzymes using chorismate as the precursor is forming tryptophan. So the precursor is chorismate and the product is the tryptophan. Now, what is interesting to note over here is that you have a tryptophan A, B, C, D, and E. This enzyme is responsible for basically the final step where indole 3-glycerol phosphate is converted to tryptophan. Now, in all these cases, what has been observed is that the enzymes are responsible for forming certain intermediates which allow it to move from one, or rather, it forms a product that is a substrate for the next enzyme that gives rise to a product that is the substrate for the next enzyme. So it is basically forming a multi-enzyme complex that is responsible for synthesis of tryptophan. So like many of the biosynthetic pathways, this pathway is also constituted of a multi-enzyme complex system. And so basically the entire operon system is giving rise to these enzymes that are able to synthesize the L-tryptophan. So we will look at the structure and the function of the tryptophan operon per se. So considering that the tryptophan operon is present within the E. coli genome and is stretched to a certain number of base pairs, it has 5 structural genes as just mentioned. You have TRPE giving rise to the enzyme TRPE, TRPD, C, B, and A. And just before and after these structural genes are present a small area which are called as spacer regions. And just preceding the spacer region is another gene of the tryptophan operon, which is TRPL. And this TRPL is preceded by the promoter, which is overlapping with the operator region. Now, there are lots of reports that suggest that there may be more than one operator region present. In fact, two half sites in an operator region is definitely something that has been reported, but many studies have also shown that in certain bacteria, you also have three operator sequences present. The promoter, therefore, is influenced by uh, a molecule that can come and bind to the operator region. Nevertheless, an RNA pol can bind to the promoter over here and synthesize this at one stretch, thereby it being a polycystron, of which this part gives rise to the mRNA that gives rise to the enzymes, and these enzymes are responsible for converting chorismate into tryptophan. So therefore, these are basically the structural genes. The TRPL gives rise to a short polypeptide which is called as a leader peptide, and this leader peptide has a role to play in regulation. In fact, the entire region from promoter till the TRPE has a role to play, has a regulatory role to play, but the TRPL has a role to play post initiation. So in this session, we are not going to look at the functioning or the working of the leader peptide or the significance of having the leader peptide in the operon, because we are looking in this session only at control at the initiation, transcription initiation. The leader peptide has a role to play in regulation, but it is post transcription. So we will take this up in a different session. And upstream, at a certain distance away, is present another set of genes which is called as tryptophan repressor and its own promoter. Now, this promoter is such that it is constitutive, it is not regulated, and one will observe that tryptophan repressor at all times is being formed from the TRPR gene. Now, when it is formed, it is basically synthesized as an aporepressor. And as we know, the aporepressor is in an inactive state. So it cannot come, its conformation is such that it cannot come and bind to the operator region. And so as an aporepressor, so when it is synthesized as an aporepressor, it is not able to bind to the operator region. Now let us look at the structure of the tryptophan repressor itself. So what has been studied through a lot of structural chemistry studies is that the tryptophan repressor is primarily a dimer. So as you can see, this is one subunit in red and this is the other subunit in red. And in fact, the entire structure is made up of, entire one monomer is made up of about 4 to 5 helices. And this also has a very typical helix turn helix complex, and it is this helix turn helix complex that is a motif that is responsible for binding to the DNA at the operator region. However, in this conformation where it is not bound to the tryptophan, it is as an aporepressor not able to bind to the DNA. But when you have the tryptophan bound at its pocket, where you have the binding site for tryptophan, what immediately happens is that there is a change in the conformation of the two subunits, that is the monomers, change their conformation such that the recognition helix of the helix turn helix is able to interact through the major groove. You can see this is the major groove, and through the major groove, it can interact with the sequence at the operator region. Same with the other subunit, you have the recognition helix being able to interact with the operator region through the major helix. And this portion of the repressor is not able to bind to the narrow groove at all. It doesn't have a very strong interaction with the narrow groove. Now, interestingly, what has been stated by many studies is that the binding of the repressor to the operator region is such that at the center, that is at the center of symmetry, you don't have the binding of the, or you do not really have the half sides present. The half sides are present more towards the right and the left of this central symmetric portion. So unlike that in the lac operon, where you have the half sides present just adjacent to each other, separated by only one nucleotide base pair, in this case there are several nucleotide base pairs that separate the two sequences to which you can have the recognition helix binding. So that is one thing that has been reported. The second thing that is important to note over here is this, that you may have more than one operator sequence present, but one doesn't know exactly how with the third operator you have the lac repressor binding, the tryptophan repressor binding. Although many studies have suggested that you have the tryptophan repressor also existing as a tetramer, and so therefore it could bind to other operator regions for the tryptophan operon itself. Now, in this case, what is very clear therefore is that the recognition helices on binding to the operator region, their interaction with the operator region becomes very strong. So it is only in the presence of the tryptophan that it is able to bind to the operator region, but in the absence of the tryptophan, it is not able to bind to the operator region. Now, let's consider the scenario where there are low levels of tryptophan. So as E. coli is growing in a media, what can happen is that tryptophan as an amino acid is required in the structure of many polypeptides, okay, and therefore tryptophan can be taken up from the media, okay, so as an exogenous source it can be taken up, or when the exogenous store becomes less, then the prokaryote or the bacteria has the ability to synthesize the tryptophan to meet the demands of tryptophan that are required to be in the structures of the proteins formed in the cell. So let us consider that there are low levels of tryptophan, which means the exogenous level of tryptophan is very less. In such a scenario, what has been observed is that the tryptophan repressor that is formed is present as an aporepressor, and as an aporepressor, because the levels of tryptophan are low, you don't have the tryptophan coming and binding to the aporepressor, which has a binding site for tryptophan. So in such a scenario, where you do not have the tryptophan binding to the aporepressor is not able to bind to the operator region with affinity, or it cannot interact with the operator region. And since it cannot interact with the operator region, the promoter and the operator are accessible to the RNA polymerase. So the RNA polymerase will come and bind to the promoter region and initiate the synthesis of the structural genes right from the leader peptide to the tryptophan A. So therefore, you will have the entire mRNA being formed as a single unit, okay, and from that you would have the ribosomes binding to the mRNA to form the leader peptide as a single unit, and then the remaining enzymes also as single units. So you will have each of the enzymes formed from a single mRNA that is formed. Because you have the RNA pol able to bind to the promoter region and initiate synthesis, slowly, gradually, the tryptophan level will increase within the cell. But originally, you definitely had less charged tRNA for tryptophan, and hence that would result in delay of synthesis of those proteins which have tryptophan in their structure. But it is interesting to note that within the tryptophan operon itself, there are two genes that do not have or that do not form polypeptides which have a tryptophan, not even a single tryptophan in their structure. So TRPE and TRPA can be preferentially translated by the ribosomes, as they do not need to wait for a charged tRNA for tryptophan. And so the TRPE and the TRPA are preferentially formed, while the other structural genes take time in getting translated, because the ribosome has to pause to have a tRNA with tryptophan come and bind, and that will happen only as and when you have more and more tryptophan being formed by the tryptophan operon itself. So therefore this fact is very interesting. Now, whether this really has a implication on the tryptophan operon working is not yet very clear, but this has been an observation that during tryptophan starvation, one would observe that TRPE and A are definitely preferentially formed. Now let us look at the condition where you have high levels of tryptophan. So this is a scenario where you have good amount of exogenous tryptophan. Okay, so when you have a good amount of exogenous tryptophan and it is being taken up by the bacteria, then of course the scenario will be that the tryptophan repressor, which is okay, and of course you would have an overall high number of charged tRNA with tryptophan. So you will not find any delay in the synthesis of any of the proteins, because the ribosome does not have to wait for a charged tRNA with tryptophan to come and get incorporated in the polypeptide being formed. So this is an observation that is there with high levels of tryptophan. So the aporepressor that is formed will immediately be bound to by the tryptophan molecules because you have an excess. Why do you have an excess? Because all charged tRNAs already are acylated with the tryptophan. Over and above that, the tryptophan, if it is surplus, it can come and bind to the aporepressor. So when it comes and binds to the aporepressor, one knows that the binding of the tryptophan will change the conformation. So you would have it changing the conformation. So as you can see, this was the conformation before the tryptophan bound. And when the tryptophan bound to it, then you can see that the conformation is changing. So this is now what is called as a holorepressor, and the aporepressor was, because it was bound to by tryptophan as a corepressor, it is forming the holorepressor. Now, understandably, therefore, the conformational change that has been initiated by the binding of the tryptophan results in a conformation that enables the helix turn helix present in both these domains to interact with the operator sequences. And because you have the holorepressor bound to the operator region, the RNA pol does not get accessibility to bind to the promoter region to initiate transcription, and hence transcription will not occur. So this is going to remain, or this is going to be how the tryptophan operon is going to be switched off until you have again a low level of tryptophan. Once you have a low level of tryptophan, these tryptophan molecules will be taken up rather for binding to the tRNA to make a charged tRNA, okay, with tryptophan. So then again what is going to happen is that these tryptophan not being present, it will result into formation of aporepressor. Aporepressor does not have affinity for the operator region, so it will leave the operator region, and now the RNA pol will again find access to the promoter sequence and give rise to transcription again. So therefore, this switched off mode of the tryptophan will continue until the tryptophan levels are high. Once the tryptophan levels become low again, you will have the tryptophan operon on working mode again. So basically what we understand is that normally the tryptophan operon is working mode. It is only when you have high levels of tryptophan present that it is switched off. So in a way one can say that the tryptophan is controlling its own synthesis by inhibiting the formation of the enzymes that are giving rise to tryptophan. So this phenomenon is what is understood as repression. That means that you have an operon being switched off, and this is an important phenomenon of the regulation of gene expression. So therefore let us look at the conclusions. The tryptophan operon has genes that enable constituting the biosynthetic pathway for tryptophan, which is switched on and off depending on the concentration of tryptophan. The tryptophan repressor, primarily a dimer, is synthesized as aporepressor, in which conformation cannot bind. That means in the aporepressor conformation, it cannot bind to the operator region of the operon, thereby keeping the operon functional. Tryptophan aporepressor only on binding to the corepressor, which is TR, the tryptophan itself, and this has to be in high quantity to bind to the TRP aporepressor. On binding to the TRP aporepressor, it forms the holorepressor that can bind to the operator with high affinity. And this is responsible for RNA polymerase not being able to bind to the promoter to initiate transcription. Therefore, it is an operon that is an example for control by repression, with the tryptophan operon giving rise to an 80-fold repression. That is the efficiency with which it is able to work. This tryptophan operon is a paradigm for the phenomenon of repression and regulation of gene expression, wherein the tryptophan itself is able to control its synthesis as it acts as a corepressor. What is also interesting is that this phenomenon of the tryptophan controlling its own synthesis, and the control is a negative control, it is akin to what is called as the feedback mechanism at the translation level. What has been observed is that when you have high levels of tryptophan present, tryptophan can also go and bind to tryptophan synthase, the enzyme, and inhibit the enzyme. So thereby you don't have any more tryptophan formation. So you can see the control is not only at the transcription level, but the control can also be seen at the translation level by the tryptophan itself. Thank you.